Just very excited that Sarah's here. So Sarah's, of course, a friend and a colleague of many people here. Sarah said she was going to be in town for CVPR and offered to swing by, and I'm really glad that she did, and I'm really glad that she brought a lot of her, or some of her lab. It's nice to meet you guys face to face after talking to some of you virtually this past year. I'm also grateful to the CVPR organizers for scheduling their meeting the same time as our EAI seminar that it worked out this year. So Sara is going to talk about a new initiative that she's been working, or a new project that she's been working on that's highly translational. And I think for a lot of us who are thinking about foundation models and sort of the limitations of existing foundation models, this is extremely relevant work, and it's very timely as well for a lot of the projects that we're talking about right now. I think that in general, the big competitions, the ImageNet datasets and the COCOs and all those, they've obviously been a spark of innovation and deep learning, and they've carried us far. But I think we're clearly seeing limitations of these datasets, especially when you think about how they actually work in real world applications. And Sarah's work, of course, is highly translational and impact oriented. Like many of our work here, and I think it's very fitting and extremely timely that Sarah's talking about this with us. So I'm thrilled that she's here. Sarah's career, of course, is highly fitting for exactly this type of translational work. So she did her PhD at Caltech and worked at Google on a major wildlife computer vision project that many people have used and is being used all around the world. And I'm really thrilled. I think we're going to learn a lot from your talk. And so thank you so much for coming here. It's great to see you. Really, I'm thrilled. So thank you so much.
Awesome. Thanks, Patrick. Yeah, so I'm happy for this to be very casual. Please interrupt me. Yeah, I think from my perspective, it's more just fun to sort of get to share some ideas about like the ways that me and some of my lab mates have been thinking about bringing and kind of adapting and expanding on existing technologies to try to actually serve some of these large-scale impact-driven scientific needs, climate assessment, and like information needs. And specifically in the biodiversity space, which is like where I've done a lot of my work. So this is sort of just one specific and quite recent project, which recently submitted this. I'm happy also, when we're sort of discussing at the end, to sort of talk about some of the other things we've been working on too. But this talk is really just focused on this one project that's really about how do we start moving towards letting scientists make better use of data that they've already collected. So data collection on the ground is expensive. It's actually probably the most expensive sort of factor. I mean, it's very cheap to sort of look at things from the air, relatively. It's very hard to collect data on the ground. Increasingly, things like iNaturalist let us collect data on the ground at really large scales. But we're really just scratching the surface in terms of what information we're really extracting from those pixels. Like we have like species ID of one of the species in the image. There's a lot more information there. So how do we kind of get at that like wealth of knowledge, and how do we think about doing it systematically? So I know there's a lot of people who work in the climate space at AI2, and there's a couple of people who work kind of in the conservation or biodiversity space. I've worked for a long time with some of the folks at Earth Ranger. But just to provide a little bit of context, biodiversity is in catastrophic decline. I think many of us are aware of this. We're seeing exponential species loss across every branch of the taxonomic tree. And the populations of wildlife have declined by almost 70% since 1970. So there might be a couple people in this room who were alive in 1970. In your lifetime, we've lost 70% of all the wildlife on Earth. That's shocking, right? And of course, this is sort of shocking and bad generally, but it's also, biodiversity is really tied to a lot of different dimensions of things that are important to human society. There's been some recent work that shows that biodiversity is tied to climate change, and that climate change obviously is one of the big factors and anthropogenic change that are leading or driving at least hypothetically, this loss of biodiversity. But it goes the other way too. So it turns out that biodiversity is actually a really strong driver of things like carbon sequestration, both at a large scale, things like elephants actually being forest managers, but also at much smaller scales. Biodiversity is really linked to this idea of bioturbation that takes forest carbon and turns it into soil carbon. When you think about the longevity of carbon ecosystems, forest carbon, 100-year roughly lifetime, soil carbon can be sequestered for up to 1,000 years. So there's like these other complicated factors when you think about these really complex, really sort of non-linear systems. Biodiversity is tied to public health in a couple of different dimensions. A really obvious one is the spread of zoonotic disease. As we're seeing wildlife populations get fragmented, they have less genetic diversity, they're under stress, they're more susceptible to disease. As we're seeing more sort of spread of human space, we're getting a lot more interaction between humans and wildlife at the boundaries, and this leads to the spread of zoonotic disease into the human population. And there's actually some really amazing work going on, like, for example, the Carey Institute, a woman named Barbara Hahn, that's explicitly studying how you can use iNaturalist data and some of the secondary information that I'm going to be talking about in iNaturalist data to predict zoonotic disease outbreaks, looking at flowering of specific species of trees that bring in fruit bats that then spread disease to the population. So there's like really sort of cool signals that you might be able to extract here. Biodiversity is tied to food security, not just pollinating our food, but actually diversity of pollinators has been shown to increase the nutrition in our food. So it's not just that we can live with the monoculture of the non-native bumblebee as our sort of global pollinator. We actually need to maintain the biodiversity of pollinators. And finally, there's just a ton of ecosystem services that the natural world provides. Air quality filtering being a big one. Stormwater runoff, soil stability. So this is clearly an important problem, and I'm not the only person who thinks that. So there's been a huge increase in interest, and as hardware has gotten cheaper, we've seen a massive increase in both the quantity and the diversity of types of data that people are collecting globally to study ecosystems and even specifically to study ecology and biodiversity. So just in our lab, we work with satellite imagery, across sort of spectral bands. We think about things like optical, but also maybe LiDAR. We haven't really actively worked on it, but I think SAR is a very interesting dimension. UAV data, much more high resolution, low flying aircraft or drone data, particularly in the sense of looking at wildlife abundance or counting, but also many other things. On animal sensors, how do you think about understanding behavior and behavior change from on animal tracks? These might be collars. I've worked for 13 years on camera traps. So this is static networks of cameras that are often motion triggered. They capture plants, wildlife, but are mostly geared towards mid to large terrestrial mammals. But increasingly, these types of camera traps are being deployed underwater. They're developing novel camera traps to study small animals. David Rolnick at McGill has been building a very large system for insect monitoring, insect camera traps. Bioacoustic sensors, static networks of sonar, for in-river fish estimation. Actually, tomorrow, a couple of my students and I are going to National Marine Fisheries, and we have a project here with NOAA where we've been working with Alaska, Washington, Oregon, and California on sustainable fisheries by doing escapement estimation, counting the amount of fish going upstream in rivers. And increasingly, community science data. So raise your hands who uses iNaturalist in this one, who's heard of iNaturalist? Okay, great. This is a friendly crowd. Yeah, iNaturalist is a really amazing story when you think about the ways that passionate community members can contribute to scientific and policy understanding. iNaturalist data is now about 97% of all of the current and historical species observation records in the Global Biodiversity Information Facility. So it is now 97% of all of the data we have on species on Earth. And it's really that's only happened in the last, 10 years. So this has now outstripped every other form of data collection pretty much that we have. And it's all just pictures that are being taken by often non-experts. We're just going out, taking photos of things that they see in their environment. But of course, it's not systematically sampled. It's incredibly biased. People take pictures of stuff they like that looks pretty to them. So you get no photos of plants when they're not flowering. A lot of photos of plants when they're flowering. It's really, really hard. One of my students, Julia, we're working on trying to do fine-grained, morphologically accurate generative data for basically improving training for rare species. It's so hard to get these generative models to generate female birds. People only take pictures of male birds because those are the ones that have the prettier feathers. So a lot of weird biases in that data. And of course, it's only really collected where people can get to. So all of these types of data, they're very diverse. They all have sort of their different complementarities, their different biases, you know, everything from 3D point clouds, there's just tons of different stuff going on here, DNA, microscopy data. None of this is a direct measurement of the things that we actually care about, right? So I want to understand animal population change. I want to understand species distribution change. I want to understand just alpha diversity. How many unique species are there in different places on Earth? This is all proxy data. So unlike some of the other climate problems that are incredibly interesting, where we want to study weather or temperature, you can put out sensors that are direct measurement of the thing you want to solve. Here, we have to collect proxy data, and then we have to process and interpret that proxy data to be able to even start doing scientific analysis. And this is really where AI has shown to have a huge role to play, you know, both in the processing and in the analysis of the data, and increasingly sort of in the unification of processing and analysis. How do you kind of do this end to end? So for this talk in particular, I'm going to be focusing on that 97% of all the species data on Earth we have in iNaturalist, these community science observations. So these are just sort of a set of random examples of iNaturalist data. And iNaturalist, this was from Friday, so I'm sure the number's gone up, but there's about 192 million observations over 477,000 species. Of course, this issue is incredibly long-tailed. So there's hundreds of thousands of images of bald eagles. People really like to take photos of bald eagles. There are lots of species that are really in the long tail. There might be 1 or 3 observations. And of course, there are many species. There are many more than 477,000 species on Earth. So there's a lot of species that have not ever been captured. But when you actually look at iNaturalist data, like I was saying at the beginning, this image is labeled by the crowd, and it's labeled plain zebra. So what we have is a species label, a crowd-generated and verified and sort of agreed upon species label for every image. But there's a lot of stuff going on here that is not captured by the labeled plain zebra. How many zebras are there? Are they zebras that we've seen before in other data? Can you do re-identification? What's the behavior? There's clearly some sort of fighting going on. Is this mating behavior? Is it fighting over food? You also are seeing species co-occurrence, and not just wildlife species co-occurrence. This is plain zebra that are grazing simultaneously with livestock, cattle. You get images of what is the amount of forage available at that place and time in the landscape. You know, what is the availability of food for ungulates or, you know, equids? And then information about, you know, much more easy to identify data of maybe the trees in the background. It's much easier to ID the species of trees from a side level image like this with the morphology, with all that higher resolution than it is from satellite imagery. So you can imagine that if you could just build something that would just let you look for the data you're interested in from this huge repository, and it would accurately be able to pull out queries, open-ended queries, that there is a huge amount of scientific analysis that we could do beyond just looking at the distribution of species occurrence. So things like intersections between humans and wildlife, in the sense of, you know, oh, these are, can you find hermit crabs using plastic waste as their shells? Or can you find animals working around man-made infrastructure, like roads? And so we're really of the opinion, and this is work that's mostly been led by my student Eddie, and it's in collaboration with some folks from University of Edinburgh, UMass Amherst, with iNaturalist itself, UCL. How do we build these interactive, expert-driven AI query systems that can provide evidence for ecological hypothesis? So as a first step, because often when you're doing what we do, which is trying to ask new questions in the AI space. You need to figure out how to measure performance. We've been building a benchmark that is trying to move towards open-ended, expert-driven scientific query. So we're calling this benchmark Inquire. And so as a first point, we've built sort of the next iNaturalist dataset. There have been sort of several previous datasets from the iNaturalist repository that have been sort of curated for the machine learning community, initially focusing on imbalanced fine-grained species ID. We had a Newt benchmark that was one of the really first steps in this direction, which is like, how do you build representation learning that then is easily fine-tunable to diverse downstream tasks? And now we're moving to this, sort of more open-ended, query-based, retrieval-based system. So first, we started by building what we call iNaturalist 2024. This is sort of in development still, so these numbers are maybe subject to change, but for now, it's about 5 million images curated from 10,000 different species classes. And then we have no image overlap from previous iNav datasets. It's filtered out and specifically split in time. So this is nice because it means that you can sort of treat these as somewhat independent datasets. They're over the same species as iNat 2021, which is about 2.7 million images dataset that we put out a couple of years ago. So, use that as you will. But the nice thing here is you could train models on iNat 2021 and you can use them on this data without sort of worrying about data poisoning. It has pretty diverse geospatial coverage. So of course, you'll note that this is not uniform geospatial coverage, but it is sampled pretty diversely from where we have iNaturalist samples. Should have added the sort of heat map of sort of global iNaturalist observations here. This is one of the sort of common and known challenges in biodiversity and ecosystem monitoring. We almost always have a lot more data in some places than others, right? And actually, really where there's a lot of biodiversity is places like the Amazon, where we have very little data. But we really sort of sought to have global geospatial coverage from iNaturalist 2024, as global as can be had from within what's sampled in iNaturalist. And then what we did is we worked with expert ecologists across disciplines. So ornithologists, forest scientists, marine biologists, a huge gamut, to try to understand what types of scientific questions they would like to be able to answer from this data, but are currently unable to. And then we've collected now and curated from those over 200 queries, and that number is growing. And then we comprehensively labeled those queries over those 5 million images. Now, there's a bit of an asterisk here where we comprehensively labeled them in some cases using things like species filters that might not be an infallible system, but we've done some analysis to try to test the sort of robustness of our, how many of the positive images we were able to capture, and we're pretty convinced that we've done a pretty good job here. And again, across all of these different experts and across all of these different things, we tried to get diversity in the types of these open-ended queries. So this is kind of a sort of a higher and then even a lower level taxonomy of the types of scientific questions that people would like to ask. Everything from health and disease, tracking identification, animal structures and habitats, cooperative and social behaviors, mating, courtship, reproduction, miscellaneous context. That could be things like finding a really high quality image of something. Like this is useful sometimes, but might not actually, the model that's good at species ID might not be good at understanding what types of images humans like. So just a couple examples of what these things might look like. One query is find California condors tagged with a green 26. So here, this is just like, examples of what you get back if you just do like simple, maybe clip-based query. You sometimes, these models struggle with compositionality. They'll give you a condor with a green tag, right? This sort of specific fine-grained details can be challenging. I think this is kind of what we're hoping this benchmark will surface. It's like, what are the things that are still challenging for these vision language models? And then, things like, I think the other nice point here is that we do get positive examples as well. So if you wanted to now take the retrievals and then filter them as an expert, you're still saving an enormous amount of time. So it's this kind of idea that you're working with imperfect AI models, and maybe we can make the AI models better, but we also can surface tools that already enable scientists to make progress and have impact where they can be using their time effectively. Other examples, Moorish geckos with regenerated tails, things like everted osmeterium, which is basically like specific defensive organs for the larvae of a specific swallowtail butterfly. I mean, a lot of this stuff is stuff that I'd never even heard of. Godwit performing distal rhicokinesis. Anyway, it's where they have this very special beak where the top of the beak can bend upward, and they can like control this. This is something that some scientist was trying to study, right? And so you want to see, like, this is one with the beak's open, but it's not doing that distal I'm not going to say it again. Redwood trees with fire scars. You can imagine people who are sort of trying to look at historical legacies of fire would be very interested in finding all of these different examples of redwood trees with fire scars. We really tried to get somewhat equal coverage across taxonomic groups, again, working within what's already exists and what's already sort of available in iNaturalist. So we have coverage from everything to mammals to arachnids. And the positive queries, so the return sort of positive examples from these different diverse queries also have quite a lot of geospatial coverage. So we're sort of not just getting queries that are maybe of interest to American scientists, for example. So simple initial benchmarks of this, try just, something like a simple clip-based query. So here you have some flavor of multimodal embedding model. So something where you're training using like a clip objective, maybe a SIGLIP objective, one of these other types of objectives, a joint embedding across images and text. So that can be trained in lots of different ways. Often it's trained contrastively, but the idea is you want some sort of metric space, some sort of embedding space where similar semantic concepts are placed close together. So then what you do is now you take that model, like this is literally just if you take clip, and you can generate embedding features for every single image in your data set. Of course, this is a little bit expensive, not horribly expensive, but you only have to do it once. With these types of models, they're very efficient because you only have to do it once. So you extract embeddings for this 5 million images one time, but now they're cached. And then for any new query, you do a similar thing, use the same model to embed that query, and then you just look at similarity in that embedding space, and you can use that to then re-rank and find the things that this model at least thinks are most semantically close to that query. And so now, okay, maybe you get a couple top matches that are actually sort of correct. And these models are, across many, many different applications, proving to be very, very successful. And we're seeing that as well. Like they do actually do a much better job than sort of things that we had previously on sort of very, very diverse stuff. But it's not perfect, right? And often these models do fail. So ways that they fail, they might have some relevant results with low precision. They particularly struggle, we found, with things like compositionality, where you'll get a high score if you match on or, not on and. And then you also, these models aren't necessarily good at scientific lingo. So this is something that's interesting. We might want to build models that then learn how to speak scientific language. But another approach that might be less training focused is just helping ecologists learn how to translate their scientific language to language types of prompts that actually are more easily understood by these models. And, depending, some combination of the two might actually be more effective. So one thing that we have found is that CLIP is pretty good at surfacing a lot of examples, but it might be noisy. But then what you can do, and still maintaining a lot of efficiency, is you can take the top retrievals from CLIP, and then you can use a much more expensive, like large vision language model, something that's actually taking in the images and the prompts simultaneously and needing to run a forward pass on those both. And that can actually keep some of that efficiency and really increase the accuracy of some of these queries. Because these large models, because they kind of have this attentive value, it's not just sort of a generic embedding, it's actually sort of some sort of embedding that thinks about the similarities specifically in this context, maybe. You do get re-ranking that's quite effective. And this actually matches what's been seen in the natural language processing literature. There's been a lot of progress in the idea of training re-rankers for NLP. And it's something that hasn't been as deeply explored in vision, but I think this seems to point to that as a very sort of rich avenue of research. So LVLM re-ranking kind of does really surface a lot more relevant retrievals, is what we found. So what we do with this benchmark is we try to actually look at both of those challenges and sort of support that. So we have a full retrieval challenge, which you go from your 5 million image data set, you do whatever you want and return your top K. But there's also kind of this more focused challenge that just says from a set of images, how do you build a re-ranking model that improves sort of the top K accuracy? And so just some of the like preliminary results we have from testing many different existing approaches. Training data has a very strong effect on model performance in this context. And it's actually quite interesting. So BioClip, which was one of the best student papers at CVPR this year, is a model that's explicitly trained. It's a CLIP model that's explicitly trained for the tree of life. It's been trained on iNaturalist data, that iNaturalist 2021 data set, and trained to have context about the taxonomic tree. But it turns out in that fine tuning, they're maintaining pretty good accuracy on species, and definitely better accuracy on species than just like the standard OpenAI CLIP model by a bit. But that fine tuning has lost a lot of that open-ended capacity of the model. So you've really lost your ability to understand behavior, context, appearance. And going from sort of the standard CLIP training and bringing in DFN, this like, filtering approach to try to get higher quality training data for CLIP. Same architecture, just different data for training. This data, this model does pretty clearly outperform on every dimension. So high quality training data seems to have a big effect. And it's not sort of sufficient to just fine tune a model on iNaturalist without, on just the species information, without sort of some way of maintaining, not catastrophically forgetting all of that other context for this problem setting.
Sorry, what is DFN again? It's what model is?
I think it's called dynamic filtering networks.
Okay.
Correct?
It's data filtering networks.
Data filtering networks, yeah. So it's basically, it's an approach that basically uses a model to decide whether or not data should be used for training.
And then a different model is trained with the filter data set?
Exactly. So then you sort of, there's different approaches. One simple thing is you just use the DFN to decide which data to use for training, and then you just train standard CLIP.
So this was all on like the test set from your new data set?
Yeah.
Each of these are trained on separate.
Yeah. So these are all existing open source models trained on separate data sets.
And which OpenAI model is
this? Is the standard CLIP.
Okay.
Yeah. They're all mid-pieces.
Yeah.
Okay.
Yeah. So keeping architecture fixed, just looking at the effect of what the different training sets are. Kind of similarly, if you keep the training set fixed and you explore changing the architecture, like many things, bigger tends to be better. We do get better performance from larger models, but you'll note that our average precision at 50, like we're not near 100, right? Like there's sort of a long way to go still. So scale alone might not be enough. And actually to really emphasize that a bit more, if we really sort of scan over, increasing computational cost versus that accuracy, you can really see that we're starting to get a pretty major plateau. So, if you put this in a log scale, we would have to kind of 10x again, sort of the scale of the model to be able to get, the sort of same amount of improvement in performance.
Is that all on the same size data set?
Yeah, so these,
okay.
Let me make sure that's actually true. These aren't all the same. This isn't all necessarily. So this is DFN, these ones are not.
Okay.
So it's not all the same training data necessarily.
Okay.
Yeah, but this is sort of just looking at like teraflops of use. And you can see that you're getting diminishing returns with scale. But of course, it's not a perfect comparison because it isn't fixed training data.
Yeah, but I guess like as you increase the training data set, the bigger models will have more capacity. So that curve might look very different depending on which data sets you use, and like
yes.
more challenging data set that scale, like the amount you increase scale, you could still see a lot larger returns.
Yes, totally. And there's a few different ways you can imagine making improvements, right? Like there's you can increase scale of models, like capacity of models. You can increase sort of the value of the training data, and you could also think about increasing sort of the value of the training signal. So like, what are the ways that you're actually thinking about training these models? And so if we look sort of across that taxonomy of different tasks, you can see that some types of tasks seem to be consistently harder than others. Nothing's really a slam dunk. I mean, even just species ID is not like solved by these models. Species ID is hard, right? I mean, giraffe versus elephant is easy, but when you're talking about like two different species of moth that look very similar, this isn't sort of an easy problem. But then definitely we're starting to see this performance go down as you think about like behavior, context, appearance. It seems like life cycle and development are some sort of very hard problems for these models. You know, what are the age or the or the sex of species? That's a hard thing. These models aren't necessarily good enough.
How are these like difficulties correlated to like the data set size and balance?
So I mean, it's so like this is looking at kind of the best in class for a couple different approaches. So this VIT-L14 with DFN, this is kind of the best sort of standard clip model. The SIGLIP model here, it's like a slightly different training objective than CLIP. That actually is also doing quite well. This is now a much bigger DFN model, and then this one is kind of like that CLIP-based plus re-ranking from GPT-4o. And we did a lot of different re-ranking studies. GPT-4o does serve as the best re-ranker, and so that's why it's sort of included here. And so these do not all have the same training data. And clearly, some of these things, we don't know what the prior is, right? Like we don't know what the training distribution of GPT is.
So I guess to rephrase my question, I was just trying to get an understanding of like, okay, depending on what the data set is for each of these different tasks, like which tasks are actually hard because they're like a really hard task, and which ones do we just have like one example of the species or like so few that have a specific disease that the challenge is actually we don't have a good enough data set, and which of them are like, oh, it's actually just a really challenging task and we need to develop new ways.
Yeah, so the metrics we use try to kind of normalize for the number of positive examples that are returned. So you shouldn't see like a massive difference. I mean, maybe if there's like very few positives, you might see some difference. And actually, that's something we should maybe look at sketching out, like performance versus number of relevant positives. Maybe we have those plots somewhere. But number of relevant positives in our data set is not necessarily going to be the same as how well represented in the prior of these foundation models it is. And that's often a much harder thing to get at. Yeah, it can be very difficult to understand, particularly as you're thinking about these sort of complex compositional things. Like, how often have you seen eating? How often have you seen eating in the context of this other stuff? Like eating, like a lion eating versus something else. Like, so I think it's a little difficult to really quantitate.
You can't know those original data sets. You don't know, like, is it actually because there's like this weird amount of overlap between like what was in OpenAI's training data sets and like what's in your test data set.
Yeah.
So there's like, I guess there's kind of, it's hard to get a clear answer of like which tasks are actually difficult.
So I mean, it's kind of interesting here. Like, it's very possible that we don't know what they've scraped. They could have scraped this data from iNaturalist. It's on the internet, but we know that the labels that we've collected from experts are not available, right? Like this is not something, it's not like easy to know like that, you know, beak bending behavior thing for these images. So there's some amount that it can't really have been scraped. Yeah. Did you have a question?
So that was just a comment that I was going to make. Like, conceivably, iNat is in all of the pre-training data, but like if they're doing a text to the image, then it's probably just the species level. Yeah,
the only information they would have is species. Yeah, but that's why I think it's so interesting that we're not at 100% for species, right? Like it's clearly like sort of still, even though that could very much be in the training data, it's still sort of not perfect. And then I think just the last kind of point here is like from a use perspective, like it's kind of impossible to think about doing like comprehensive, like, you know, large vision language model, you know, query, a specific query for every image. Even on 5 million images on an A100, doing some like LVLM ranking could be something like on the order of 57 days per query. Clearly not happening. You could make this faster with parallelization, but of course, then you need a lot more GPUs. Clip zero shot for 5 million images, if you just do some like nice, like sort of nearest neighbor tricks, can be like a second, right? So this is so fast, so interactive. You get a big boost from that combination, but still it's like 2 minutes. And so I think there's another sort of dimension of research here, which is thinking about like efficiency as a goal. So how do you kind of maintain accuracy, but really increase the efficiency of this type of query? Because we're only talking about 5 million images here, but you imagine you're scaling up to 197 million images, it'll sort of get that efficiency again becomes more of a challenge. And not every ecologist has an A100. So, whether we want to have this be central or we want to sort of open source different models and enable people to do this more generally, I think is interesting. And iNaturalist we're working on sort of releasing a demo sort of publicly with iNaturalist, just to start, also kind of getting more feedback about these types of tools from the community. And yeah, part of that is, and sort of how we collected these labels is we built an API. The royal we, Eddie built an API that really makes it easy for people to do this efficiently, do this filtering efficiently. It's really sort of working with like the parallelization of the human visual system, right? It's very easy for us to search through grids of things and do pattern matching. And so then you can, expand the image if you need to look at fine-grained details. But so it's kind of fast in this way to think about filtering and finding which ones actually are correct from the ones that are returned. And this is kind of ready for use in a way. Like it's not sort of perfect, there's room for improvement, but it's already valuable. And we've gotten a lot of sort of enthusiasm and interest from different ecologists in the community. I mean, specific things like we met one ecologist in Denmark recently that was, they've been building custom apps for community science sort of approaches to try to just explicitly find pictures of jellyfish on beaches. It's so easy to just say jellyfish on beach in this place with something like this. Like there's definitely sort of a lot of low-hanging fruit here, just in terms of like things that people are putting a lot of investment in to try to answer. But the challenge is specifically what types of questions is this type of system good for? So we can very reasonably and very impactfully answer find me examples of type of questions. But being able to say something systematic about find me every example of is still, I think, really difficult to guarantee. And so this kind of gets at this kind of expanded goal, I would say I have, which is like, how do we move beyond just returning the images? And how do we think about actually returning analysis and answering questions over sets of images? So the example I keep bringing up is like, what if I want to say like, tell me the distribution of species of flowers that hummingbirds are eating in the Pacific Northwest. So you find all the examples, so something like a bit more maybe like Viper GPT style, but instead of image captioning, it's more answering questions about sets of data. And I think the real cool thing would be bringing in some of that ecological statistics to start thinking about what is our certainty on the types of questions that we're answering based on the data we're pulling from, based on the samples. Yeah.